Okay, so good evening and good morning, everyone. Welcome to the 11th dialogue of the Breathe series. We are excited about today's topic on cognitive health. We are absolutely thrilled to have Dr. Sandra Dedesko and Dr. Sarah Haines with us today. These are the two most respected voices in the indoor air quality and built environment space, and we are really grateful to have them share their insights with us today. Before we dive into today's session, a quick introduction for those who are joining us for the first time. The Breathe In series is organized by us at Environmental Design Solution. My name is Dipanjali, and I'm joined by my colleagues Lakshmi Nidhi and Tanmay. The Breathe In series is a platform where we talk about indoor air quality, everything about indoor air quality, why it matters, the challenges around it, and most importantly, what we can do about it. The topics we've explored have taken shape around four key themes. One is regulatory convergence, then it's design and technology, monitoring and management, and creating awareness among occupants. So, we've had the privilege of learning from experts across the globe covering a wide range of topics. We've looked at everything from indoor air quality standards, monitoring and performance frameworks to emerging technologies. And we've explored how indoor air quality plays out across different building types as well, like residential spaces, schools, healthcare settings, and office buildings. That brings us to right into today's topic. We often define good buildings as aesthetic, cooler in summer, warmer in winter, or maybe some good lighting and decent acoustics. But how do people react in these buildings? There's a quote that I'd like to refer from Sir Winston Churchill that really captures this idea. We shape our buildings, thereafter, they shape us. It's actually a powerful thought, and it's something experts are now observing and validating through data, research, and building science. From the materials we use to how we ventilate and maintain our spaces, buildings actively influence our overall cognitive performance. We generally would refer to indoor air quality if there's a strong smell or when a room feels stuffy. As experts, when we talk about designing spaces or assessing for good indoor air quality, we have to consider interactions between a lot of indoor air quality parameters. So, one widely used way to group these parameters is particulate matter, gaseous pollutants, and bioaerosols. Indoor air quality is often linked to sick building syndrome, and we have covered it in our previous dialogue. However, certain indoor air quality parameters may affect cognitive functioning, memory, reaction time, mood, and the whole range of human functioning. So I'd like to give you a quick example. We did a real-time monitoring study in Delhi. For instance, here we are looking at CO2 levels spike at night in a bedroom with closed windows and probably no fresh air intake. We are talking about 2000 to 2500 ppm and beyond. So, long-term exposures of such levels could lead to issues like headache or fatigue, and so on. This is data from a kitchen apartment in another building in Delhi. The TVOCs, or the total volatile organic compounds in a kitchen, are another red flag, indicating, probably cooking as a major indoor source of TVOCs. And apart from cooking, TVOCs could also hint at, something deeper. For example, hidden mold or dampness that could affect our cognitive functioning and overall health and well-being. So monitoring and managing these indoor air quality parameters is therefore at utmost important. Well, indoor air quality is actually starting to receive formal attention around the world. Some countries already have well-developed indoor air quality guidelines in place, while in others, the process is still evolving with growing momentum. certain pollutants like, if you can see in this chart, there's particulate matter, carbon dioxide, and other VOCs have been studied extensively and are increasingly being regulated. But other microbial pollutants like bacteria and fungi still lack standardized framework or clear thresholds in many regions. And that's why we need more deeper research, better choices in building materials, and smarter and innovative building designs. So therefore, these are the things that will not only keep indoor air quality within safe limit, but actually create indoor environments that support long-term health and well-being. That's so there's a lot to uncover in this space today, and that's exactly we'll explore with our speakers. So they bring some of the most current research and practical insights on microbial exposure, indoor air dynamics, and their impact on human health and cognition. We would love to make this session more engaging and interactive, so and learn from you all as well, from the participants as well. So please feel free to use the Q&A function to ask questions and share your experiences. We will also be launching a few polls during each session, so do keep an eye on the pop-ups on your screen. With that, let me introduce to Dr. Sandra Dedesko. So Sandra works at the intersection of engineering and public health to improve the health and sustainability impacts of buildings. With a background in civil engineering and building design, Sandra pursued a PhD in public health to better incorporate health into her work. She's now a scientist at the Smart Living Lab at EPFL in Switzerland. We are honored to have you on our Breathe In Dialogues. Over to you, Sandra.
Well, I'm honored to be here, and thank you so much for the opportunity. Hopefully everyone can see that. But thank you again for that really generous introduction and for the opportunity to speak with you all and for everybody attending. So today we're going to be talking about designing spaces for improved cognitive performance. That's my part. And then Dr. Sarah Haines will be talking about her research on the indoor microbiome. And to give you a little bit more context about where I'm coming from, Deepanjali gave you an overview that I do have a background in civil engineering, both my bachelor's and master's at the University of Toronto. And then I did spend quite a bit of time in building design consulting work where I had this growing desire to better incorporate health into my work. And that's what ultimately led me to pursue a PhD in public health, still focused on building design. And now here I am at EPFL trying to combine both backgrounds in civil engineering and public health for more balanced solutions to our built environment challenges. Today's topic is really going to focus on the research from my PhD dissertation, which is all around the impacts of indoor air quality on cognitive performance. And it really is going to be a blend of both engineering and public health. So if you are from more the engineering or the architectural side, some concepts might be new and same vice versa. So if you do have any questions, feel free to pop them in the chat and I'll try to answer them during Sarah's presentation and then also during the live Q&A afterwards. So as I said, today's discussion, at least my part, is really focused on linking the built environment to different cognitive impacts. And first, I'll start with a brief introduction and background on this topic. Then I'll launch into the main part, which is new CogFX insights. CogFX was the study series from my PhD work. And then finally, I'll wrap it up. What does it actually mean? I know sometimes academic research can be hard to translate, so we'll try to walk through what it means in practice and go through some solutions that could have benefits beyond just cognitive performance. And before I dive into the contents of the presentation, I do want to start with some acknowledgements from my time at the University of Toronto with Dr. Jeff Siegel during my master's, then at the Harvard School of Public Health with Dr. Joseph Allen, and now at EPFL with Dr. Dushan Lachina. I've learned so much from all of these teams, both the advisors and my fellow teammates. So the contents of this presentation wouldn't be possible without working with all of them. And I do have to give a special shout out to the Harvard Healthy Buildings team led by Dr. Joseph Allen, since pretty much all of today's research will be from that team. So past and present members deserve full credit too. So starting on with topic one, what is the background on indoor air quality and cognitive performance? And I think Deepanjali highlighted this really nicely in the introduction, that I feel we really have had this growing understanding That started with a lot of concerns about outdoor air pollution, possibly also because it's often more tangible. We can like see and sense outdoor air pollution and its sources more readily than those indoors. But over recent years, we've started to become more concerned about the impacts of different indoor air exposures, with another common example being cooking, something that is more sensory oriented. But I am curious, and we can test out our first attempt at a poll question. Which are you more concerned about for your health? Is it typically outdoor air quality or indoor air quality? And I'm also curious, since I know our audience members are going to cover different geographies, so I am curious to hear. Okay, wow, very interesting that more than half of us are more concerned about our indoor air exposures or the indoor air quality. And I assume maybe our audience isn't fully relatable to the broader audience, because probably a lot of us know that since we spend most of our time indoors, our indoor exposures are governed by our time indoors. So, okay, very cool. We're already a very motivated group on this topic. And then this is a more recent, familiar, I think, exposures, impacts that are more tangible. But here on the right, I have a seemingly kind of healthy, normal office building. And this is a more recent line of research on the impacts of cognitive performance in different spaces. And that really is the topic of today's talk. I am, once again, though, curious to know at this time what you are more concerned about on a typical basis from air quality. Is it impacts on your physical health or more so on your cognitive health? Okay, so it seems pretty close, but slightly more people responded that they are more concerned about air quality impacts on their physical health over cognitive. And so we will actually end today's discussion touching on both of those points, even though the talk is focused on cognitive research. Oops, sorry, I gave too much away. And so on this topic, even though I think traditionally a lot of us have been focused on the physical health impacts, there is this growing body of research examining the cognitive impacts of air quality on our health. So this is a summary of the CogFX, that's short for cognitive function, research series that was launched in Dr. Joseph Allen's lab at Harvard, where they first started with one study that was called CogFX Indoor Environmental Quality, and that was largely focused in a controlled, simulated office type environment. Then there was study 2, which was buildingomics, looking at kind of green building factors and their impacts on human health. And then finally, there was study 3 with global buildings. And to give you just a quick snapshot of some of those study findings, so this is from the first study that took place in that simulated office environment, where they took a small group of office workers and exposed them to different conditions, where they experimentally altered different conditions related to ventilation and material selection. So they just categorized these as conventional, just say a typical building, typical ventilation, no healthy material selection. And then they went to different levels of like health certification or better indoor air quality. And so to keep things simple, we'll just say that from going from conventional to green, that's one step to better indoor air quality. And then to green plus, that's another step. And then they measured people's cognitive performance across these different domains, from more simple things like basic activity level, focused activity level, all the way to more complex domains. So like the type of strategy you're using, your breadth of approach. And what they consistently see across these different domains is that as you move from the conventional to the green, and then from the green to the green plus, you see this measured improvement in cognitive performance related to these building design decisions that improve indoor air quality. So this was a very cool study. I was still working in consulting at the time, and I was captivated by this study. Of course, time that they did a cognitive test, and then their cognitive test scores. And so what you can see is as CO2 concentrations decrease, we see an increase in the levels of accuracy of the cognitive tests. And this is even over a very low exposure concentration range, all the way down to what would theoretically be outdoor or atmospheric levels. So one of the key findings, as you can see, is there is this continuous improvement or this linear, no threshold effect, as we would say scientifically. So they found this continuously improved score in cognitive performance with lower CO2 concentrations. And then building across this, my dissertation research in the team built upon studies 1, 2, and 3 by incorporating a study on creativity from the global building study, and then also launching a separate study examining schools. So to kick things off with global creativity, an overview, we were examining associations between indoor air quality and something called divergent creative thinking, which is basically a type of creativity where there's no right answer. It's about coming up with creative solutions to a problem. Why we wanted to study this was to examine less studied cognitive processes, as a lot of research is on more objective measures like simple math problems, information usage, these types of things. And we also wanted to incorporate more diverse geographies using the global buildings data set. And so you already had a quick snapshot, but it's basically the same base data set that we used to launch this creativity analysis. And this time, from those monitors at the participant workstations, we focused mostly on carbon dioxide concentrations and fine particulate matter concentrations. Unfortunately, the volatile organic compound concentrations, that's a type of gaseous pollutant metric, there are many challenges with that, from low-cost sensors, both the measurements themselves and then interpreting the measurements for health. So unfortunately, we don't consider them useful for this type of modeling work. So we focused on carbon dioxide and fine particulate matter, those very small particles in our models, and then also incorporated temperature and relative humidity. In terms of actually recording the cognitive test scores, everything is done through a custom-built smartphone research app where we send participants something called the Alternative Uses Test, AUT, where basically they're prompted with an everyday object, so Band-Aid, and then they have 2 minutes to come up with as many distinct uses for that as they can. So something to cover blisters, arts and crafts, the list can go on. You can get some wild responses. And then from those raw scores, we calculate 4 metrics. Fluency, that's the number of unique solutions they came up with, so in this case 2. Elaboration, that is the level of detail, which we quantify as the number of words used. Flexibility is the number of distinct use categories that we have to define manually. And then originality, these are the number of distinct uses when we compare the same prompt across all participant responses who had that prompt. So 4 different metrics. Don't worry about the details of them. They should all correlate with fluency, and many believe fluency to be the most important one in terms of this divergent creative thinking metric. So from all of the possible responses, we had 618 responses to the AUT test, and unfortunately, through a data cleaning and scoring exercise, many of them had to be removed for incompleteness or a lack of validity. And then unfortunately, the sensor technology was not as advanced at that time, so many were missing exposure data, so we had to exclude those. So overall, we had just under 200 tests from 86 participants from 4 countries, including India. Unfortunately, we did not have data from China and Mexico. And just to give you an idea of the indoor conditions, here are some summary statistics for what we measured and included in the models. You can see overall that the mean conditions are really, I would say, quite low and in line with what you would expect from buildings operating to standards, say from ASHRAE. And then it's really when we look at the 95th percentile values, we're starting to see a higher exposure range, but overall they are still quite low compared to the range that is possible. And so overall, these buildings are quite well operated and maintained. Then to try to model the relationships between indoor air quality and the scores, we use a type of statistical model that is really flexible and allows for many things that traditional regression does not. And then we control for several variables in the model that could confound or cause error in our model dissociation. And basically, we always want to choose the model with the lowest number of parameters. So this is the ones highlighted in blue that had the best fit. And this is really just more for the intense scientific crowd, but we also provided a fully adjusted model just for anyone curious about other variables that could confound or bias the model dissociation. And so Before I move into the results, I want to clarify a few things that often come up, and we can chat more during the Q&A. But the first is you may have heard people say that correlation doesn't mean causation. Well, association also does not mean causation. So everything I'm presenting today, these are modeled associations. We can't make inferences past when this goes up, this goes down. It's not saying that caused that. And so this is one classic example shown here where we can see chocolate consumption on the x-axis and Nobel laureates per population on the y-axis. And so it would seem that chocolate consumption causes Nobel Prize recipient, but that is not the case. It's just a correlation. It's not causal, unfortunately, for everyone. And then another question that often comes up is, what does CO2 actually represent in these models? So in public health, this is a commonly used tool. This is a directed acyclic graph, but basically it allows us to draw out our study scenario. And basically what you're seeing here, this is something called an indirect effect, where this variable x acts on y through a different variable z, but x also acts on y directly right here. And so this is a common debate in the indoor air quality field where we have CO2, and it seems to be associated, not causal, but associated with cognitive function in some way. And so far, there are many differing opinions, but so far I haven't seen anyone quantify this direct or indirect effect through something else. So whenever I'm referring to the results with respect to CO2, we can't actually say what the mechanism is. So just keep that in mind. And so back to the results. This is a summary of the different effect estimates between our indoor air pollutants and our creativity scores for both those minimally adjusted and fully adjusted models. And what I'd like to draw your overall attention to is that we see this consistent pattern that when CO2 concentrations increase by 100 parts per million, we see a decrease in AUT scores. And in most cases, these reach statistical significance. When we move on to PM 2.5, we see a similar trend that an increase in PM 2.5 is associated with a decrease in cognitive scores. However, you will see that they don't reach that statistical significance. So we say they are suggestive. This is suggestive evidence. And this is in line with past work that has only found that significant effect at concentrations higher than what we observed in this study. So these results aren't to say that particles don't matter. They likely do, but it's often at concentrations higher or at data sets more powerful than we had in this study. And so the key takeaways from this study is that lower indoor CO2 and PM 2.5 concentrations are associated with this improved creativity scores, even over a short period of time and a low exposure range. So I think that's really key to remember that we were looking at really short-term exposure times and a low exposure range. Next, moving on to the classroom study. This time we're looking at indoor CO2 in classrooms with higher ventilation rates for COVID-19 and metrics of accuracy and speed, so not creativity. Why we're interested in investigating this is to understand the potential benefits of increased outdoor ventilation rates, which were prescribed for COVID-19, beyond just infection risk reduction. And how we did this was once again with our very flexible statistical models, but this time we launched a whole separate study. Where we followed many graduate students at a Boston-based university over an academic year. And we installed continuous environmental monitors in classrooms where they were regularly attending class. And then we engaged with them once again with our smartphone research application, sending them a series of tests and surveys after class. And so you can see we had a live stream of various environmental measurements. We used temperature, humidity, and CO2 in our models. And then for the outcome data this time, we had one of two cognitive tests. This is the Stroop test that measures your cognitive speed and inhibitory control. So it's your tendency to want to select dominant information, but you have to refrain from doing that and select the less dominant information. So it's kind of like an impulse control. It's tricky, but it is fun, that test. And then I think this one is more boring, but it's also more familiar. It's simple addition and subtraction. And then we also had a survey question that allowed us to ascertain if the student was actually in their class exposed to the concentrations that we were measuring. This time we had much more data, just over 800 cognitive test responses from 71 participants. Unfortunately, though, a lot of students didn't complete the paired survey, so we didn't know if they were in class. And then those that did, a lot of them indicated they weren't in class, so that is not great for our study. So we ended up with just under 300 cognitive tests from 54 participants. And to give you an idea of the exposure range, this is actually even lower than in our last study. You can see really low carbon dioxide concentrations, a really tight temperature range, and low relative humidity, although this occurred over the heating season in Boston, so it's not necessarily surprising. And then once again, we used this very flexible statistical model to control for our exposures and potential confounders. And to show you the results, this time we're looking at peak classroom CO2 exposures on these different cognitive metrics. So on the top, we have measures that incorporate the number of correct responses per time. And on the bottom, we just have response time. So there are 6 different metrics available from these 3 different tests. This is Stroop, all of the responses on the right. This is a subset of the Stroop responses that are more difficult to complete. And on the right, we have arithmetic. And what I'd like to draw your attention to is that when we see these increases in indoor CO2 peak exposures, we see a consistent decrease in correct responses per time. And we also see an increase in response time. And most of these reach statistical significance. And so we do see this overall trend that with increasing CO2 concentrations, we see this consistent decrease in cognitive function scores. And so the key takeaways here are that lower CO2 concentrations, which in this study were achieved by changes in ventilation and occupancy, are associated with improved cognitive test scores. And this is a really key finding because in schools where occupancy is typically fixed, and we saw from the pandemic that in-person learning is a real priority for student development, outdoor ventilation rates could help lower CO2 concentrations or something else that might be important and support student learning. However, I do want to highlight that this is a situation where outdoor pollution was low and it was quite feasible to implement outdoor ventilation. And so tying it all together, I know that's a lot of scientific content, but what does this actually mean for practice and how should we act on it? So the summary of findings, I've basically said that it seems when we have better indoor air quality, we have better cognitive performance. But there are many, many limitations from these studies, largely related to the generalizability of them, or how we can take the results from the study and apply them more broadly. And so 2 that I really want to highlight when we're trying to think of solutions is that a lot of these study situations involved low outdoor air pollution, or at least low indoor exposure ranges, because so many of the buildings were so well operated and maintained. Another challenge is that since these are just association models, we can't point to causality. So we don't actually know which indoor air pollutant it is that is causing these observed cognitive effects. So I would say, like, the takeaway, improve indoor air quality. And you're probably going to say, but how? So whole question here, what are your biggest barriers to improving indoor air quality? Is it knowledge on how to do it? Is it resources, cost and available materials, behavioral change, say maybe not wanting to do it? And I don't mean that in a negative way. That could be something, for example, like changing the way you cook. And I find a lot of the cooking methods that result in the best tasting food often also result in the worst indoor air pollution. So that's something I struggle with. Is it outdoor air pollution, or is it a limitation of the building? Say there's no opportunity to implement like a particle filter. You don't even know how to access it, these types of things. Okay, so it seems quite split. A lot of it is resources, but it seems spread across the board that there are many challenges, and I would agree. But let me walk you through a few possible solutions, and hopefully this will help cover many areas. So first of all, just to reiterate, even though a lot of the results are based on CO2, that's more about convenience for studies, and so we're not necessarily saying it is CO2 you need to target. It could be something else, something related to something that's harder to measure, or we don't even know to measure, which I know Sarah will talk about more. But for now, it seems that even when we improve things related to CO2, other things in the building improve also related to indoor air quality. So let's talk about a few general strategies we could use to improve indoor air quality. following what is called the hierarchy of IAQ strategies. So that's first trying to control sources, not introducing things into the space that could degrade indoor air quality. Then trying to provide adequate ventilation to dilute pollutants, and then providing targeted air cleaning, such as filtration or portable air cleaners. And so to give you an example, by selecting healthier materials, so this was a furniture study done by the Harvard Healthy Buildings team, where they saw that selecting healthier furniture and carpet reported to be PFAS-free had lower median levels of organic fluorine, known as a proxy for overall known and unknown PFAS content. So they found a measurable difference and then went on to analyze dust samples and found lower hormonal activity. So a measurable benefit of healthier materials. And you can also expand this to even healthier purchasing with the products like fragrance-free products that you would use at home. In terms of ventilation, I know we all have limited ability to actually change the ventilation system, but if you have even a range hood at home, like over the stove to cook with, turning it on and ideally high will, as you can see, help reduce particle concentrations. If you don't even have that, like I've had that problem in many apartments, you can try to open a window and use a fan to exhaust air out that way. Another question in terms of using outdoor air pollution, are you concerned about ventilation, outdoor air ventilation due to outdoor air pollution? Yes, no, or unsure. Okay, most people are concerned about outdoor air ventilation, which I understand. So let's talk about other strategies like filtration, these media filters that can help filter out particles. And the higher the rating, the more effective they are at reducing particles of various sizes. So here's an example again from the Harvard team where they looked at PM 2.5 concentrations both outdoors and indoors. And you can see it's variable, much higher in countries like China and India. But you can see also that the particle filtration, especially the higher rated ones, did quite a good job at reducing indoor concentrations overall. And then finally, does your home have particle filtration? Yes, no, or unsure. Okay, I'm not surprised. Most people also said their home does not have particle filtration. This is quite common. So I did want to end by highlighting that even if you don't have a place to actually put one of those big filters in your ventilation system, you can use standalone portable air cleaners or air purifiers. You can buy commercially available ones or make your own by combining one or more filters with a fan to get that particle filtration in more localized areas. And I do want to end because I know a lot of people said they were more concerned about the physical health impacts, and I totally understand. A lot of the solutions I just discussed that are, targeting better indoor air quality and can provide cognitive performance, there are so many benefits beyond that. So we know with higher air pollution, we see decreases in survival, and with higher ambient particle concentrations, we see increases in death by many causes. We also see an increase in risks for other cognitive ill health conditions like dementia, Alzheimer's, or even decreases in height. And so I think there are broader benefits of particle filtration than just cognitive performance. We can see life expectancy decreases for population-weighted particle performance. And we can see that in these countries, at the higher end, there could be greater impact from reducing our exposures. And this graph here on the right is showing current life expectancy decreases from particle exposure, but on the bottom, we're seeing improvements in life expectancy if we were able to lower particle concentration. So I know this presentation was largely focused on cognitive performance, and whether you are more motivated after to improve indoor air quality for cognitive performance, I want to really emphasize that there are physical health benefits to improving indoor air quality. So whether you do it for one or the other, you will get impacts for both. So happy to take any questions you have in the Q&A, and I also look forward to the live Q&A session. So thank you, everyone.
Thank you so much, Sandra, for sharing your research and experiments. Yeah, as you rightly said, cognitive performance, creativity and productivity, it becomes really important for certain settings like schools and offices as well. So thank you so much. And for any questions for Sandra, I will encourage all of you to use the Q&A function. We will also take up some questions during the moderate session in the end. So I'll just quickly move on to our next speaker. Yeah, so we have Dr. Haines, who runs the indoor microbiology and environmental exposures lab at the University of Toronto. She is also a founding member of the Hub for Advancing Buildings. Dr. Haines holds a PhD in environmental science, where her research particularly focuses on the impact the built environment has on human health. She uses cutting edge microbiology techniques to understand the impact of moisture on indoor environment quality. So that's an interesting topic. So over to you, Sarah.
Hey, thanks so much, everyone. Happy to be here. Just going to share my screen. There we go. Okay. Hopefully you can all see that. Okay. Okay, well, yeah, thanks, everyone. I'm Sarah Haines. I'm going to be talking about exploring this intersection of indoor microbiomes and health. So Dr. Dedesko gave us a great kind of overview on this intersection between, thinking about your cognitive function and this piece of our health, and then, how air quality is impacting it. I'm going to focus more on our microbiomes and how all of these different things in our indoor spaces could be impacting health. I'm going to really focus a lot on respiratory health in that interaction. So as was mentioned, I run the Indoor Microbiology and Environmental Exposures Lab at U of T. So some of you may have, and hopefully you did, come to the previous Breathe In dialogue with Professor Jeffrey Siegel. I work also with Professor Siegel in the Hub for Advancing Buildings, where we're really looking at this interdisciplinary research, looking at how we can improve human health and comfort, promote equity and resilience, and carbon emissions in buildings, and all these different types of buildings in our built environment at U of T. So just to get into it, what is the indoor microbiome? And I like to think of it as this kind of, I say, somewhat invisible world all around us, because, sometimes we can see microbes. And by that, I mean, right, maybe you've seen active mold growth on some building materials. I mean, hopefully not, but I think we probably all have at some point. And our microbiome is really made up of all of these different components. So bacteria, fungi or mold, and then viruses. And a lot of our bacteria in our indoor spaces comes from people. So we actually populate our indoor spaces with our own microbiome, which I'm going to talk about. And then a lot of our fungi and mold actually come from outdoor sources, which I'll get into as well. But then, you could have plants that are going to be sources, and their soil, as well as your cats and dogs and other animals, right? So we have lots of different sources of microbes in our indoor spaces. So our first poll question I'll bring up is, what sources of indoor microbes are you most concerned about indoors? Okay, all right. So we see a lot on mold and moisture, right? So a lot of you are concerned about mold in your indoor environment. So I mean, it's not a good thing, right? To be worried about mold and have mold in your home, but we're going to talk about it. So it's a good thing that you're here today. We see also people, animals, right? Plant, soil. I'm actually going to talk about all of these things and how all these different sources of indoor microbes could impact health, but maybe could have some beneficial impacts as well. So we're going to get into that. Okay, so coming back to kind of these different types of microbiomes. So as I mentioned, right, our bacteria on our human body actually populates a lot of our indoor spaces. So the human microbiome, it consists of all this different microbiota that relies on or, you know, inside or on our bodies, on our skin and in our gut. So maybe you've heard of, right, this gut-brain axis. There's been a big push for keeping your gut healthy. It keeps your body healthy. So that gut microbiome, just like that, we have this human microbiome that's populating our spaces. And I know some of us, you know, we're engineers and building scientists, and this might seem, oh, I don't know about all this, having all this bacteria on my body, but it's good for you overall. And we really influence our indoor microbiomes, right? And as I was saying, we're populating these spaces. And so there's been lots of different work done that has really found that no two home microbiomes are the same. So in the study by Lax et al., they actually found that as folks were moving from different houses, so let's say they sampled your home microbiome, but then you moved to a new house. They actually found that in a relatively short period of time, only a couple days, they started to see the home microbiome of that new house started to look very similar from that previous house, right? So we are actually, you know, populating these spaces, bringing in our microbes and changing these environments. The same was found in a study that I was involved in where we were looking at fungal community composition, and we were seeing that actually when we looked at different homes, so on that figure on the right, it looks kind of crazy, I know, but every single color is a different home. And then in these kind of plots, similar species, so similar types of fungal species cluster together, and you'll start to see some clustering of those different dots. And that was really showing that, again, there were similar fungal communities within each house. And this was really interesting because we were actually looking to see if there was any relationship between exposure to high moisture conditions. And we found that that didn't really matter. It was all came down to the different homes, right? So similar fungal communities were clustering together. So your home fungal community and bacterial community is different than mine, is different than someone else's, which I find very fascinating. So there's really this bi-directional relationship, right, between the human microbiome and our indoor microbiome. And I think it, you know, there was a quote at the beginning by Winston Churchill saying, you know, we shape our buildings, they shape us. And we're really finding that with microbiomes as well, that there is this relationships where we're introducing our host-associated microbes, but then the built environment may be introducing microbes or introducing different contaminants or impacting our health in this way, right? So we're kind of having this bi-directional relationship with our indoor spaces. And coming back to health, there really is this link between these bioaerosol exposures and respiratory health. And normally in presentations, I don't like to have, you know, lots of text and like a table like this, but I found this very helpful because it kind of breaks down, right, these different potential exposures. So things like allergens, dampness and mold, which you all said, you know, this is something you're concerned about, bacteria, viruses. And thinking about this link to health and respiratory health, especially where we're seeing a lot of these different exposures, could then cause, you know, asthma exacerbations, or could cause asthma in young children, cause wheezing and other diseases. And then thinking about how we can control it, right? With some of these control strategies, we see source removal, maybe it is moisture control, and some filtration, which I'll get to, and some ventilation. So to start with mold, because I know you all said you were concerned about mold. Building dampness and mold is related to respiratory health. And we see in many different studies, there is this link between being exposed to building dampness and mold and then having adverse health outcomes, especially asthma or respiratory health. And I know 2007, it's a little dated, a statistic, but in the US, you know, they were finding that many cases of asthma were actually related directly and attributed to dampness and mold. And so especially in young children, we see this relationship. So with that, and maybe it's less of a poll, I'm going to quiz all of you. What is the ideal relative humidity of an indoor environment? What do we think? Okay, let's see. Results are in. Great. You guys know your stuff. You've seen indoor air quality talks before. Yeah, 30 to 50%. So, you know, keeping things too dry, you can end up with other health impacts, right? So maybe your eyes and nose and throat kind of dry out. You could get nosebleeds. Your skin feels very dry. So you don't want things too dry. So you don't want that 10 to 20, but you don't want it too humid, right? 60 to 80 or even above 90, you're going to start to see that mold growth. So that 30 to 50% is really that ideal space that you want to keep your home environment. And we actually see that in with some of the work that I've done, where we've looked at microbial growth in different building materials. And we actually see that as you increase the equilibrium relative humidity, so that's on that x-axis there, so we increase the humidity, we start to see that microbial growth, that mold growth, that's there on the y-axis. And so the first figure is actually in carpets, and there's that nice fun photo on the right where you can actually see a mold growing on a carpet square. I know it's kind of gross. I find it very, very interesting, but we actually see that mold growing. And the same for drywall. Really, when we get above about 85%, that's when you start to see this mold growth. So actually these increased levels of humidity, we do start to see mold growing on our building materials, which is why, right, we want to keep things between that 30 and 50%. And not only can increasing the relative humidity increase just the actual growth, but we also see that it's associated with increasing allergen concentration. So this work was done by Dr. Nick Nastasi at Ohio State, where they were looking at, this specific Alt-A1 allergen. So this is a fungal allergen. It is comes from Alternaria alternata, which is a common indoor fungal species. We start to see, again, as we're increasing this humidity, potentially increasing these allergen concentrations, which could lead to these poor health outcomes, right? There's this relationship, as I mentioned, where we're seeing high mold, high dampness, maybe release of allergens could be related to poor respiratory health. Okay, so we know that we can have these harmful health outcomes, but what about these potential beneficial impacts of different microbial exposure indoors? And so some work has actually been looking into, finding that exposure to a diverse set of fungi indoors maybe is actually beneficial to respiratory health. So no, you don't want to be exposed, mold and dampness is related to asthma outcomes, but they're actually finding that maybe being exposed to a diverse set of fungi in your built environment, not active mold, not dampness, right? But just having diverse fungal microorganisms in your home maybe could be beneficial to respiratory health. And we see that in this figure here, where you can see asthma cases in purple on the bottom, and then the control, so no asthma in your home, on the top. And the ones on the top there in the control, they actually have more fungal OTUs, which would be more diverse, different types of fungal, so more different types of fungal species in these homes. And not only that, but we kind of in the field took this a step further and started to look at, okay, what about farm-like microbiota? So what kind of other microorganisms might we need to be exposed to kind of prevent asthma? And we're seeing, again, and this is all a lot of similar to what Dr. Dedesko was saying, right? Where we're seeing these associations, we're seeing these relationships. I can't say for certain, right? I'm not going to say, oh, you need to be exposed to farm-like microbiomes, but we're seeing kind of these associations where when you have farm-like different types of bacteria, we're actually seeing, less likely that young children in the first, I believe it was 6 years of life, they were less likely to have asthma. So they may be protected, right? So being exposed to these certain organisms maybe could protect you from getting these harmful respiratory outcomes. And I had it highlighted. So there you go. In the bacteria specifically, not as much in the fungi and for this one, but farm-like bacteria. So maybe it's that soil microbes that could have protective effects against asthma. And not only that, but certain yeast maybe have protective effects against certain allergic airway diseases. And this yeast, Vishneocosmia victoriae, is actually associated with dogs. And so potentially having a dog that has this yeast maybe could reduce your child's risk of asthma. And so actually There are, I know some folks who have actually, they have a baby and they got a dog to see if it would, be helpful in preventing asthma. So that's that interesting relationship. Again, I'm not saying to go out and get a dog. I'm not, there's still more research that needs to be done, but it's this current kind of research that I found very fascinating where we may, be looking to engineer these microbiomes. So my question to you all is, should we be engineering our indoor microbiomes to promote beneficial microorganisms and influence human health? Okay, so people say yes, right? We're saying yes. We should engineer our microbiomes to promote health. We see some no's, we see some maybes, don't know, right? And I think that's totally fair because it is a little, sometimes when I talk about this it sounds a little sci-fi, right? Where maybe we're going to promote certain beneficial microorganisms in our indoor environment, but what could that lead to, right? What happens when we're promoting certain microorganisms, and how could that impact health overall for many different people, and what does that do to other microbiomes? So I think we need to be really thoughtful, right, about how we're going to influence our spaces, because yes, there could be these really great positive benefits, but also there's some, potential negative outcomes, which we talked about at first. And I think we still need to determine what is a healthy built environment and what is a beneficial microbe, right? Because I think the idea, and you also, many of you said, yes, like we should be doing this. But before we do that, we do need to really determine what is a beneficial microbe. And we're seeing, right? Maybe it's this exposure to a diverse set of fungus. Maybe it's this outdoor farm bacteria. Maybe it's dog fungi or associated yeasts. But we still need more research on that, right? So how do we really do this? Well, I'm going to start a little segue, and it'll all hopefully all come back together on plants, right? And I think when we think about healthy built environments, a lot of us often think about plants. So do indoor plants, what do you think? Do plants positively influence indoor air quality? Okay, so we're seeing a high majority of folks are saying yes. We're seeing some no's, some maybes. Okay, so many people are saying yes, they do influence indoor air quality. Well, I like plants. I'm not gonna lie. I'm not gonna say I have indoor plants, but I will say the jury's kind of out on if they are really positively impactful for indoor air quality. So some of the kind of negative impacts, right, of indoor plants is they might increase humidity and moisture, but they also may influence the presence of certain microorganisms, and some of these could be harmful, right? So when we're bringing plants indoors, as I mentioned before, with our mold and moisture, right, high associations of mold, moisture, high dampness, this is going to be related to poor respiratory outcomes. So things like plant walls, and this is a plant wall that's actually at University of Toronto. It looks beautiful, right? Aesthetically, it's beautiful, but potentially having this amount of plants, if they're not taken care of regularly, or you're bringing in lots of different soil microbes, you could potentially, bring in some harmful microorganisms, or really increase the moisture in your indoor space, right? So it's kind of this, I would say, this balance of yes, they're aesthetically pleasing, and I don't have the studies on this. I should have included them. But often we see this relationship between seeing plants and actually having, influenced mental health. So actually visually seeing plants actually boosts our mood, helps us be more productive at work. And there's actually some studies going on right now where they're looking at, does it need to be a real plant? Could it be a fake plant, and so on? And how does that influence your mood and your mental health and so on? But with plants, to actually have an influence on our indoor air quality, maybe we would need a lot of plants. So there's this idea also that plants can remove harmful volatiles, right? So you maybe have heard, oh, I have plants inside, and it's gonna reduce these different volatile organic compounds that could be in my indoor space. And that on the right there, we actually see studies that were done in 2020 by Cummings and Waring. They actually found that you would need many, many plants to actually, like 100 plants per meter squared to actually, have some kind of loss rate, to actually see some kind of air exchange, ventilation rates, and reducing those VOCs. You would need so many plants in your indoor space. And then on the left here, we actually see, could our plants influence our microbiome? And again, this was a study done by Bridget Haggerty's group at Case Western, where they found that you'd actually need so many, potentially many plants to actually change the indoor microbiome, right? So thinking back to that farm-like microbiome study, this idea of, okay, maybe we need to bring those these outdoor sources of bacteria into our indoor environments. Maybe that's not the route yet, or maybe it is, and we still just need more research. But right now, you just need a lot of plants. So potential air cleaning mechanisms on plants, they really can't compare with ventilation. And so I'm going to end my talk just talking about ventilation and filtration, where, this might be one way that we can really manage our indoor microbiomes for health. And so when we're thinking about all these different pieces that I talked about, how are we going to then engineer these spaces? How are we going to engineer our microbiomes? And maybe it is through ventilation and filtration, which can both remove harmful pollutants, but maybe could also populate some spaces. And actually, Sandra had one of these figures also in her presentation. And, with different ventilation strategies, we actually see in some work that they can influence bacterial communities. So in both these studies, the first one, we're seeing that using mechanical ventilation, or opening your windows to get ventilation, we're seeing different bacterial communities coming in. And I think that makes sense, right? If we think back to the beginning of my talk, where I was saying a lot of our bacteria, some of it comes from people, some of it comes from outdoor sources. And so with mechanical ventilation, we're seeing a lot of very human associated bacteria. So potentially, circulating throughout your home. But then opening your window, you see some more outdoor sources. So thinking about what we want to bring in to our spaces, ventilation is going to be critical. And not only that, but ventilation is also critical for managing airborne pathogens. And I didn't talk a lot about viruses in this talk. There's actually a lot of more work that needs to be explored for viruses in our indoor microbiomes. But we all just lived through the COVID-19 pandemic, and we know, right, that these bioaerosols, these airborne viruses, we need to be thinking about how to control our spaces. And again, one of those ways is going to be through ventilation. And so this work by Lydia Maraska was looking at, how can we design our spaces, right? Is it for occupancy? Is it to save energy? Maybe we need to start looking at personalized ventilation, right? Where clean air is supplied directly to a person, and thinking about how we're designing our different spaces for who's there, and what we want to see, and what we want, exposures we want to prevent, or which ones we want to see. And then air cleaners, as additionally, and Sandra also talked about this, right? And air cleaners can be used to reduce these concentrations as well. And I actually have some work being done where we're looking at, using air cleaners and filters to reduce bacteria and fungal concentrations and aerosols indoors, and actually collecting those samples onto filters, and then looking at what's there, and kind of using these filters to see what's there. But when these air cleaners are appropriately sized, and that's what's really important, right? Is they're sized correctly for a room, we can actually see these really great reductions in airborne viral concentrations. And that in the corner there is those DIY air cleaners. I should have had a poll, if anyone has built them. If you haven't, they're really easy to build. They're pretty fun to build, especially if you have kids and everyone likes putting them all together. You just need some air cleaners, a box fan, and some tape. And they actually work really, really well. So on a whole, when I asked, should we be really designing, engineering our indoor microbiomes? And if we are, here's some ideas. And I like this graphic by a new review, where we're thinking about, maybe it is increased microbial diversity exposure, right? Maybe we do need to be exposed by diverse microorganisms. Maybe it is increased ventilation. Maybe we're bringing in, you know, at some point, maybe it is microorganism enriched materials that we're bringing in. Maybe we have figured out, once we've made it to that point, we know which microbes are beneficial. Maybe we are bringing them indoors. I think we're not quite there yet. I would say we still need more work, but you know, these different pieces, I think the direction of the indoor microbiome work in this built environment space is really progressing in a great direction, and I'm excited to see where things go. So with that, some kind of closing thoughts. I think it's really important to consider the microbiome when we're thinking about our built environments. We need to be careful in considering, you know, how are we balancing these positive and negative kind of benefits of these microorganisms. And then, you know, using ventilation and filtration could be helpful tools for this management. So with that, we'll stop, and thank you so much.
Thank you so much for sharing your studies on the relationship between microbiome and health, especially respiratory health. And it's interesting to see the mold growth on carpets with increased humidity. And yeah, giving some clear strategies in designing our spaces. So now we move on to our Q&A session, and I invite Lakshmi to please moderate this session. Thank you.
Yeah, so thank you, Dipanjali. Really excited to moderate this session. To our participants, this is a great opportunity to ask questions and listen to different perspectives on the topics covered today. So I encourage all of you to type in your questions in the Q&A box. And we'll take up as many as possible today. So without further ado, let's welcome our experts and get started. So I'll start off with you, Sandra. You know, in terms of the effect of carbon dioxide on our cognitive performance, I wanted to know, how do you look at emerging technologies like continuous air quality monitoring and, you know, connected smart ventilation systems, you know, shaping the indoor air quality exposures?
Yeah, that's a great question. It does tie back into, I tried to highlight it briefly in the talk, and there's never enough time because it's a topic all on its own, but there is so much debate around CO2 and how do we interpret it. So I guess I would say, like in my studies, I certainly find CO2 useful, even if there's unanswered questions about whether it's causal, whether it really is the cause of those cognitive effects we see. It is useful for measuring. So with the emergence of these low-cost sensors, it's very practical to measure the CO2 concentrations for the studies. And then even in the introduction where you showed, you know, you can easily do a sleep study. Well, sorry, I don't know what the monitor uses, but you can, even at your own home, you can do your own study around your house and see how CO2 is increasing. And you'll often see that the CO2 is correlated with other air pollutants that I hinted at, that they're trickier to measure. So again, like lots of scientific debate around the topic, but I still think CO2 is kind of this like useful marker that can tell you about other things happening in your space. And yeah, to your other point, CO2 is also useful for ventilation management. So a lot of systems kind of modulate or change the ventilation rates based on measured CO2. So it's a very practical measurement. It doesn't paint the whole picture, but I think it is very, very useful.
Yeah, I also would like to follow up on that. You know, even though we have low-cost sensors, they are yet not accessible to a lot of people, right? So, in that case, how do you look at managing our exposures when you know, monitoring is not accessible?
That's a great question. And so you're right, like they can be so helpful. You can learn so much about your space if you do have access to one, but most of us don't. And so there are certain things, like for example, in the sleep study you showed, if you are sleeping in a very small room with the windows closed and you know it's kind of like a tighter envelope, like you don't feel drafts or anything, you can assume that the CO2 is increasing at night. Or say some other common activities in your house, like cooking. We also had some slides on cooking. That's going to generate a lot of pollutants, whether or not, sometimes people think it's just gas stoves. The actual device you're cooking and the food itself, that generates a lot of pollutants as well. So if you're cooking at home, that's a time where you want to be paying attention to opportunities to bring in outside air or try to use a fan to push the pollutants outside if you can. And then just other things, if you're using highly fragranced products, that's going to bring in these gas phase pollutants. So trying to select fragrance-free products if possible. And then whenever you're bringing something new into the space, that new smell, like that you're not imagining that is a new smell. So it's these types of consistent activities, or say, it would be remiss to not mention this, but if you're burning anything indoors, you're going to generate a lot of pollutants, whether it's a cooking, a heating device, a candle. So these types of moments are good to pay attention to. And oftentimes you can be your own sensor. Like if you smell something, if you feel irritation in your eyes or nose, chances are you're detecting something. So I would rely on yourself as a monitor too.
Right. We do have a question for you, Sarah. The question is, how far are we from the use of real-time measurements of the type and diversity of microbiomes and their beneficial roles, and how can we use bioaerosol sensors to monitor microbiome?
Yeah. Okay, that's a great question. I see it here. So
Yeah.
I would say, how far are we from the use of these real-time measurements?
Yeah.
I think we're getting there. I think, we're working towards currently right now. Like you've seen, right, this kind of change we had. We're focused on just quantitative methods, right? So qPCR, and maybe everyone remembers getting their PCR tests during COVID, so we can quantitatively get this. And now we've moved into sequencing, where we can, and even in the past 20 years, the cost of sequencing has gone down significantly. We're actually now moving more from sequencing and actually looking at, the 16S sequencing, ITS, where you're seeing, okay, what types of microorganisms are there? Actually moving towards, more of this transcriptomics, where we're thinking about what they're doing, what's their actual function. And, I don't know if I have a good sense of, how far off we are by the time, we're actually quickly taking samples and they're getting run in the lab, or maybe we have some kind of measurement tools that are just in your home. But things have been progressing very, very quickly, and I do think this field is moving fast. And so who knows? I mean, maybe in the next 20 years we'll start to see this. Something Sandra just mentioned about the smell, right? Smelling VOCs. Actually smelling mold, seeing or smelling mold currently is one of our best indicators for acknowledging finding the mold in your home or in buildings, right? So we're still kind of a ways away on, using these certain detection methods. But I think maybe using microbial VOCs, that was something that I was doing some research in. Maybe using these VOCs that the microorganisms are releasing themselves could be a detection method, right? And so I think that kind of gets to that second question of using bioaerosol sensors to help indoor microbe monitoring. Maybe it is, I think, finding these biomarkers or things that we can detect that maybe they're releasing, right? So the microorganism, the microbes are doing something and they're releasing these compounds, or something's happening that we might be able to detect. So yeah, I think hopefully we'll see it in our lifetime. Yeah.
Great. And also, as a follow-up question, what could be the probable issues surrounding the use of certain beneficial microbes? I mean, it could be different, as we said. I mean, as you had described in your presentation, it could be different for different people. So, what could be the issues when it comes to managing beneficial microbiomes, and how do we identify?
Yeah, no, it's great. Yeah, so I think, right now we have these potential associations, right, where we're seeing you can be maybe it is exposed to these outdoor microbes, or it's these soil microbes, or dogs and so on, that are helpful to some people, right? But it might not be helpful to everyone. And I think that's where we do need to pause and really think through, maybe for the general population this is helpful. So maybe the general population having a plant wall inside is beneficial. Who knows, maybe that is the route that these beneficial microorganisms. But there may be folks who right, are it's having this exposure is going to be harmful, and how do you really balance that to someone who has maybe a chronic illness or so on, and that's going to be harmful. And so I think we do need to be careful about thinking, oh, these could be good for everyone, when maybe it is more specialized, or it could be harmful to some folks, and kind of balancing that, or putting something in place where maybe you didn't know you were going to be exposed to it, right? And now it's going to have some negative health effect. And I think that comes with everything, right? There's going to be that balance of some people could be harmed in some of these things, but I think we do need to think about it.
Great. We do have another question, or maybe a thought on the carbon dioxide and its impact on productivity. So, it is seen that pollutants like PM2.5 and PM10 are very popular, but unlike CO2, so since it affects cognitive performance, shouldn't we be making people aware of the importance of CO2, especially in settings like offices and educational institutes? I mean, how could we ensure that this awareness on CO2 particularly is ensured amongst the public?
I see. And I think I see the question in the chat. So this is the kind of controversial topic in the indoor air quality cognitive performance research space, where a lot of people will say that there is no effect of CO2 on cognitive performance. So in my opinion, we still have, of course, more research to do. There are some studies that say there is an effect, and then a large group feels that it is maybe one of those indirect effects, right? Like CO2 to some type of volatile organic compound to cognitive performance. We still don't know that part. To the question about how to kind of educate and raise awareness around this, I do think the original CogFX studies provided a lot of motivation. And we did hear, I mean, I was still in design, you saw a real uptick in people wanting to do these kind of healthy building audits. I couldn't believe how much this paper was being passed around in industry. And then we did also see an uptick in the use of these low-cost continuous monitors. So I think those there was an awareness just from kind of the dissemination of this research. To the question, like, how can we raise awareness beyond cognitive performance, if I'm understanding that properly? It's tricky with CO2. There's still a lot to be understood about the different biological mechanisms. At really, really high levels, that's where we start to see more extreme health effects related to like a lack of oxygen and oxidative stress. At lower levels, you'll see something like sick building syndrome symptoms like nausea, drowsiness, this type of thing. I think it's always kind of a provocative question to ask people, like, do you feel tired in the office? These types of very tangible things where you start to get that slump in the afternoon. A lot of people believe that could be due to CO2 exposures. So I think it's tricky because there's so much more evidence for particulate matter when we see the really intense physical health impacts. So I think it's a priority, like what are the immediate health impacts, and is it an office, is it a home? How do we want to intervene? And I'm also happy to start typing. I know there's a lot of questions coming in if that works, but I don't want to,
Yeah, we do have really good engagement here. So there is another question in terms of, is there any data on the effect of ozone and VOCs on, that are known as hormone disruptors and cognitive abilities? Is there any data or study done?
Sure. So one of the earlier CogFX studies, the one in the lab that has a section on VOCs, where they experimentally altered the VOC levels and measured the impacts on cognitive performance. There's a more recent paper that just focused on VOCs and creativity as a cognitive outcome. And they found, again, higher VOC levels resulted in a decreased cognitive performance, this time in a creative domain. I'm not aware of anything looking at ozone and cognitive performance. I think that would be tricky due to the respiratory health considerations around ozone and exposing people to that intentionally. So I'm not so sure on that, but I could make a hypothesis.
Great. Yeah, this one is for you, Sarah. So the question is on the portable air filters. While they filter the particulate matter, there could be an issue of carbon dioxide buildup. So shouldn't we be looking at fresh
Yeah,
air sources?
absolutely. And I'm
Yeah,
sure Sandra can also
yeah,
comment on this. Yeah, so I wouldn't say completely, replacing all of your ventilation with just portable air cleaners, right? There's a difference between I would say ventilation and then filtration. So you still want ventilation where we are having active air movement, right? Where you're moving and moving air through different means, be it mechanical and so on. But the portable air filters are really good at filtration. So they're good at removing particles, right? They're good at removing particulates and potential other, getting dust out of the air, all these different particulates onto your filter, and potentially, removing bioaerosols, so things like viruses and bacteria and so on. They're going to get trapped on your filter. But yeah, they're not good at removing gases. That's not what they're for, right? So you still want that good balance of having filtration, but also that ventilation. I don't know if Sandra has anything to add.
No, that was great. I did want to chime in just briefly because I know the question phrased it like ventilation to bring in fresh air, and
Right.
that would be ideal. But unfortunately, like a lot of times the outdoor air isn't as clean as we would like to actually, dilute indoor pollutants. So that's why I was really pushing for, or like really promoting the filtration as, or the portable air cleaners as a strategy when you do have that outdoor air pollution challenge. But you are right. Ideally, we would have a balanced approach.
yeah, great. Again, there is another question on the beneficial microbiomes. Do we have a list of beneficial and harmful microbes? Yes.
Yeah, unfortunately, I'm not really. I would
Yeah.
say I think that's kind of where we're looking to go. I think there are ones that we know are harmful, right? There are
Mhm.
ones out there that, especially certain viruses and bacterial pathogens, right, that we know, okay, these cause harmful health effects. I think the beneficial side, we still need more work in. And again, there's these relationships that we're seeing, and potentially these could be causing these helpful outcomes, but we still don't know yet. But I'm hopeful. I think this is potentially where things could be headed. And then I know they asked exactly what we had just talked about. Some might be helpful to someone, maybe they're harmful to someone else, and I absolutely think that's something we have to consider.
It, we are about at the end of our scheduled time, so we'll probably take up maybe one or two more questions. So one is on identifying the right portable air filter. Are there some design considerations or specifications?
Yeah, I think
Yeah,
we can both probably
yeah,
comment on
exactly.
that. Yeah. Yeah, I mean, I would say, it depends on which ones what you're getting, and I would say for the most part, if you're purchasing just ones online, or you're getting them from different manufacturers, there is going to be, they will recommend, right, for this kind of room size, for this kind of space. But it really comes down to the ventilation rate as well, and that, like, how quickly like you're getting those air changes, right? What's the air change um, rates through those different devices. So I would say just, checking before you're buying something. Is it going to be for a small room? Is it built for a large room, and so on? And then looking at the different types of filters, like, you can get different MERV ratings of different filters, and you want to be careful that you didn't buy something that it's really, maybe high filter efficiency, but maybe it won't work with the HVAC system that you have, right? So maybe
Yeah,
it's not going to actually pull air through. It might be too tight
yeah.
to actually get anything through. So you do want to be really careful. I don't know, Sandra, if you have anything else on kind of
I dropped
sizing.
them.
Yeah.
I responded to the answer. I don't know if other people
Oh,
can see it,
perfect.
but the Harvard Healthy Buildings team put together a portable air cleaning sizing tool based on room size
Great.
and the air delivery rate that you would like. So if you click on the
Oh,
link, that
great.
should be user friendly. So hopefully that helps.
Perfect. Look at that.
Great.
Dual effort.
We do have, a few people asking about, additional reading materials. So probably we'll get back to you when maybe we can, create some list of reading
Yeah.
materials that we could share with our participants. So I think we have taken all the questions, and we are at our, the end of our schedule time. So, thank you so much, Sandra and Sarah. It was amazing talking to you and listening to your, work, your experience and insights. Thank you so much for sharing, those great ideas and strategies with us. And I'm sure all of us have these key takeaways to advocate in our own built environments. So before we conclude today's session, I want to thank our experts and then also thank you to our audience for your active engagement and participation. Follow us on social media to stay updated on our next dialogues. With that, we bring this webinar to a close. Thank you so much.